Assalamu alaikum dear students. I am Dr. Shahjabeen and today's lecture topic is transport of carbon dioxide. These are the lecture objectives. We will discuss the ways in which carbon dioxide is transported in the blood along with the importance of chloride shift and Haldane effect, and we will discuss the carbon dioxide dissociation curve. As discussed earlier, in the transport of oxygen, the carbon dioxide is also transported due to the constant circulation of blood throughout the body and its diffusion from higher concentration of partial pressures to the lower partial pressure gradient. But this time from the tissues to the lungs, that is in the opposite direction. Here are some partial pressure of carbon dioxide that aid in diffusion. You can see when the arterial blood enters into the cell, it has only 40 mmHg partial pressure of carbon dioxide. Intracellularly, we have 46 mmHg. Interstitial fluid and venous blood have 45 mmHg, and when it enters into the alveoli, there is 40 mmHg partial pressure of carbon dioxide. Here you can note one more thing that is different from the oxygen, that pressure differences required to move from one point to another are less than the oxygen. It is because carbon dioxide can diffuse about 20 times as rapidly as oxygen. Now we will discuss the transport of carbon dioxide from the tissue cells to the lungs in 3 steps. Number 1, diffusion of carbon dioxide from the tissue cells into the blood. Then the ways in which this carbon dioxide is transported in the blood, and in the end, the third and the last step, which is how carbon dioxide is released in the lungs, or you can say alveoli. Now come to the first step of diffusion of carbon dioxide from the tissue into the blood. Here you can see that the arterial blood that is entering into the cell, it has only 40 mmHg of partial pressure of carbon dioxide, whereas intracellularly there is 46 mmHg pressure. This is all because carbon dioxide is formed inside the cell due to continuous metabolism going on. Now look here, the partial pressure of carbon dioxide in the interstitium and the venous end of capillary is 45. So all the carbon dioxide will diffuse from cell to the venous blood. Note that although all the cells are continuously forming carbon dioxide, but the concentration gradient difference is only 1 mmHg. This is all due to the rapid diffusion quality of carbon dioxide, as I have told you before. Come to the second step of transport of carbon dioxide, that is transport of carbon dioxide in the blood. Under normal resting conditions, each 100 milliliters of blood can carry 4 milliliters of carbon dioxide, and it carries it in 3 forms. Most of it, that is 70%, is in bicarbonate form. Then 23% is in carb-amino compound form, that is in combination with hemoglobin and plasma protein. Rest 7% travels in the blood in dissolved state. A very small portion of carbon dioxide is transported in dissolved form to the lungs. The venous blood, with the partial pressure of carbon dioxide around 45 mmHg, contains 2.7 milliliter per 100 ml of carbon dioxide in dissolved state, while the arterial blood with a partial pressure of 40 mmHg contains 2.4 milliliter per 100 ml of carbon dioxide in dissolved state. So if you minus 2.4 from the 2.7, you will get 0.3 ml of carbon dioxide. Yes, only 0.3 ml of carbon dioxide is transported in a dissolved state per 100 ml of blood from tissues to the lungs. And this is about 7% of all the carbon dioxide that is transported in the blood. Most of the carbon dioxide that is transported in the blood is in the bicarbonate form, that is 70%. How this bicarbonate is formed? After entering the blood, most of the carbon dioxide that enters the RBC reacts rapidly with water to form carbonic acid. Do remember that this reaction occurs due to the presence of carbonic anhydrase in the RBC. And this carbonic anhydrase catalyzes the reaction between carbon dioxide and water and accelerate it about 5,000-fold as compared to plasma, where it takes place and it takes more time. Now this carbonic acid that has formed is a very unstable compound. So in a fraction of a second, it dissociates into bicarbonate ions and hydrogen ions. The bicarbonate ions diffuse out of the RBC into the plasma, while the H plus ions are buffered by hemoglobin protein, which is a powerful buffer and capture these H plus ion. When bicarbonate diffuses out of the RBC into the plasma, the inside of the cell becomes less negatively charged. So, in order to replace or neutralize this effect, negatively charged chloride ions diffuse from the plasma into the RBC to replace bicarbonate. This movement of chloride ions into the RBC is called chloride shift or hamburger phenomena. This process is mediated by a special bicarbonate chloride protein known as band 3 or anion exchange I protein, as it shuttles the ion in opposite direction at a very rapid speed. Although the bicarbonate that is formed within the RBCs, most of it is diffused out of it, so it is carried in the plasma as bicarbonate, and some of it remains in the RBC and transported from there as well. This is the pictorial representation of the previous lecture. Here you can see that carbon dioxide is diffusing from cell to the interstitium and then into the blood due to the partial pressure of carbon dioxide gradient. Some of it being carried in the dissolved form in the plasma, this carbon dioxide, and when it enters into the RBC, it is converted into carbonic acid under the action of an enzyme, that is carbonic anhydrase. The carbonic acid is then dissociates into H plus ion and bicarbonate ion, and these bicarbonate ions are diffusing out and chloride is coming in. What is this? This is chloride shift or hamburger phenomena. Yes. What else you can see? At the end, what's going on? Yes, Bohr's effect. What was that? We have done it in the oxygen transport in the previous lecture. Bohr's effect states that presence of carbon dioxide, here it is, presence of carbon dioxide or the H plus ion decreases the affinity of hemoglobin for oxygen. Here, this is going on. When the H plus ions that are formed after the dissociation of carbonic acid, they were captured by the hemoglobin protein, buffered by the hemoglobin protein that dissociate the oxygen from the hemoglobin, and the oxygen will dissociate out and goes into the cell. This is Bohr's effect. 23% of the carbon dioxide is transported in the form that is in combination with protein. When it is combined with the amino group of plasma protein, it is known as carb amino protein. And when it combines with the amino group of hemoglobin, it is known as carb amino hemoglobin. Here you can see when some of the carbon dioxide enters into the RBC, it will combine with the hemoglobin and form carb amino hemoglobin. This is it. This is the carbon dioxide dissociation curve. Let me take the pointer. Yes. This is the normal carbon dioxide dissociation curve, and it is obtained by plotting the relationship between partial pressure of carbon dioxide in mmHg on the x-axis against the total volume or content of carbon dioxide in volumes percent. What is volume percent? It is the amount of carbon dioxide in milliliter per 100 ml of blood or in milliliter per deciliter of blood. This graph shows the linear relationship between the carbon dioxide and carbon dioxide partial pressure and its volume content. Look at here. This is the linear relationship. But practically, in the body, the partial pressure of carbon dioxide value of arterial and the venous blood varies within 40 and 45 mmHg. In the arterial blood, 40 mmHg, while in the venous blood, 45 mmHg, which is a very narrow range. And this is the normal operating range. Secondly, this graph shows how much volume of carbon dioxide is transported in the blood. You can see 4 volume percent of carbon dioxide is exchanged during normal transport. How? As you can see, as concentration or amount of volume of carbon dioxide rises to 52. This is 50, 52 volume percent. As blood passes through the tissues. Like here, when the partial pressure of carbon dioxide is 45 mmHg in the venous blood, in the tissues, and falls to, here it falls to 48, only 48 volume percent when it passes through the lungs and it has the arterial partial pressure of carbon dioxide is 40 mmHg. So you can minus 48 from 52, that gives you 4 volume percent. 52 and this is 48, 52 and this is 48, and that gives you 4 volume percent, or you can say 4 milliliter per 100 milliliter of blood is transported. Now, an important phenomena in the transport of carbon dioxide is Haldane effect. We know the Bohr's effect that happens at tissue level, in which carbon dioxide decreases the affinity of hemoglobin for oxygen and displaces it or offload it. The reversal or opposite is known as Haldane effect, and it occurs in lungs. It is when oxygen binds with hemoglobin, it tends to displace carbon dioxide from it. Haldane effect is essential for the release of carbon dioxide from blood into the alveoli in the lungs. What is the mechanism? The mechanism is the combination of oxygen with hemoglobin in the lungs causes the hemoglobin to become a stronger acid. And this highly acidic hemoglobin has less tendency to combine with carbon dioxide to form carbaminohemoglobin that were formed by the combination of hemoglobin protein, thus displacing much of the carbon dioxide that was present in the carbamino form. And this increased acidity of the hemoglobin also causes it to release an excess of hydrogen ions that were previously buffered by the hemoglobin proteins. When these hydrogen ions are released, they bind with the bicarbonate ions to form carbonic acid again, which when dissociates into water and carbon dioxide. And this carbon dioxide is released from the blood into the alveoli and finally into the air. This graph shows the significance of Haldane effect on the transport of carbon dioxide. Here you can see two carbon dioxide dissociation curve. One for the deoxygenated blood, that is the dotted line, and below is the carbon dioxide dissociation curve for the oxygenated blood. Now look at the deoxygenated curve at point A. Partial pressure of carbon dioxide is 45 mmHg at the tissue level, and partial pressure of oxygen is 40 mmHg, you know. And what is the carbon dioxide content? It is 52 volume percent. Now look at the oxygenated curve. In the lungs, when partial pressure of oxygen is 100 mmHg and partial pressure of carbon dioxide is 40 mmHg. And now, what is the volume or amount of carbon dioxide? Yes, only 48 volume percent at point B. Why it happened? Just because of the Haldane effect. If there was no Haldane effect, go above, like here B, the 40, when the oxygen was less, 40 mmHg in the deoxygenated curve. Now, what is the content of carbon dioxide? 50 volume percent. So, what does it mean? Due to the more carbon dioxide in the oxygenated curve, or you can say in the lung, carbon dioxide content falls to 48%. Otherwise, it would be 50 volume percent. Thus, the Haldane effect doubles the amount of carbon dioxide release from blood into the lungs. Now come to the third and the last step of carbon dioxide transport, that is the release of carbon dioxide into the lungs. When the venous blood of partial pressure around 45 mmHg reaches pulmonary capillary, the diffusion of all the three forms of carbon dioxide occurs into the lungs, or you can say alveoli. Number one, the release of carbon dioxide from the carbaminohemoglobin into the plasma. As you know, when the oxygen enters into the hemoglobin in the pulmonary capillary due to the higher pressure, what happens that this oxygen combines with the hemoglobin and form oxyhemoglobin and displaces carbon dioxide from the carbamino hemoglobin. This carbon dioxide then diffuses out into the plasma. And in the second form, that is the release of carbon dioxide from bicarbonate into the plasma, same thing as discussed earlier. Oxygen displaces, when it enters into the RBC, it displaces the buffered H plus ions from the hemoglobin. This is all Haldane's effect, that the oxygen displaces H plus ion in carbon dioxide. So these H plus ions that are released increases the acidity and causes the bicarbonate from the plasma to enter into the RBC. The bicarbonate that is already present in the RBC along with that enters into the RBC. All of this bicarbonate will combine to this excess of H plus that has been released from the hemoglobin due to the combination of oxygen. And when they combine, they will form carbonic acid H2CO3. This carbonic acid will dissociate into water and carbon dioxide, and again this carbon dioxide will diffuses out into the plasma. Note one more thing, that when bicarbonate comes into the RBC due to the increased H plus ion, inside cell becomes more negative. To neutralize this, chloride return to the plasma, and this is known as chloride shift reversal. Now, in the last, the carbon dioxide that was present in the dissolved state in the plasma plus release from the carbaminohemoglobin, and that was formed due to the bicarbonate combination with H plus ion. All of these combinedly exerts partial pressure of carbon dioxide around 45 mmHg. Since the partial pressure of carbon dioxide in the alveoli is 40 mmHg, so the carbon dioxide from the plasma enters into the alveoli of the lung due to the partial pressure gradient. And from the alveoli, due to the constant ventilation, carbon dioxide is transported to the atmosphere. In the end, what is a respiratory exchange ratio? that is also known as respiratory quotient. It is the ratio of the rate of carbon dioxide excretion to the rate of oxygen consumption per minute. Normally, the rate of carbon dioxide excretion is 4 milliliter per deciliter per minute, you know it, and rate of oxygen consumption is 5 milliliter per 100 ml per minute. That gives 4 divided by 5 or 0.8. So the respiratory exchange ratio for a person on a normal diet consuming average amount of carbohydrates, fats and proteins, this would be zero point 0.8, and some of the book says 0.825.
Thank you, students. This is my email address for your queries and the reference books names.